All right, welcome back, fellow scientists. So let's review chapters 8 and 9, photosynthesis and cellular respiration. We memorized and explained the life equation for both processes. We looked at the structure of a chloroplast for photosynthesis, the structure of a mitochondria for cellular respiration. We described the two steps of photosynthesis, so that's the light-dependent reactions in the Calvin-Benson cycle. We described the three steps of cellular respiration, so that's glycolysis, Krebs, and the electron transport chain. So now we are almost done with this unit, and we are on to learning objective number 5, describe fermentation. All right, so I'm going to go ahead and grab a new page. Why don't you go ahead and turn to a new page in your notebook? That just makes the most sense. And go ahead and write down, there it is, aha, fermentation. Fermentation. I'm going to use blue because fermentation, as we'll find out, is without oxygen. And if a human being goes without oxygen for long enough, then like their lips and their face and a little bit like turn blue or purplish or just not the right color. So anyway, so here we go. A little bit of background before we actually dive into the process of fermentation. So cellular respiration requires oxygen. CR, cellular respiration, requires oxygen, requires O2, right? So if we look back at the electron transport chain, we can see, aha, right here, electron transport chain. Come on now, give me my bar. There it is. So we can see right here we have our oxygen that picks up this electron from pump 3 and gets made into water. That's where oxygen comes into this whole entire process. If we look at our overview diagram that we did, if you flip back to that in your notes, right, we can see the same thing, right, that there's no oxygen anywhere in glycolysis or Krebs, but then O2 has to come in and has to swoop in to pump 3 and pick up the electrons and then is made into water. So cellular respiration, oxygen, that's why we breathe. There's a fancy scientific word for that. It's called aerobic respiration. Oh, sorry, AE. Aerobic respiration. I don't know if anyone still does aerobics, but it was around like before Pilates, before yoga. It was a low impact, kind of exercise workout where you stayed active, but you were able to take in enough oxygen to remain active, and you didn't really like breathe heavily. It's not like sprinting or like weightlifting or running. It's aerobic, right? So you're able to take in as much oxygen as you need it, right? So cellular respiration requires oxygen. Fermentation, on the other hand, does not. Fermentation does not require O2. So it's not aerobic respiration, it is anaerobic respiration, right? If you remember GLR's Greek and Latin roots, a or an means not. So down is with O2, W and a slash means with, it's shorthand for with, O2, right? Fermentation is anaerobic. Anaerobic, and that is without. W slash O means without O2. Okay, so let's take a look at when does fermentation happen? When does this happen? And this is where I have to cheat a little bit because I don't want to spend a lot of time writing stuff. So I typed out what I want you to write down. So if you need to pause it, that's totally fine. But this happens when there's a shortage of oxygen. For example, when you're sprinting, right? And the reason why this happens when there's a shortage of oxygen is because electrons clog the pumps on the electron transport chain. So if we go back to our diagram of the electron transport chain, there we go. So you'll notice oxygen picks up this electron. Well, if oxygen isn't there, then this electron stays there. And if this electron is there, then transport protein number 2 can't take an electron over here, and then pump 2 can't get rid of its electrons, and so on and so forth. It's kind of like a traffic jam on the ETC, right? I could imagine like a traffic helicopter, like,
yeah, we got a traffic jam on the ETC. It's a parking lot down there. Just stay at home. You're not going to get to work anyways.
right? I mean, it's a traffic jam on the ETC, so it's horrible. If there's no oxygen, then electrons just clog the pumps. Well, Mr. Leo, what does that matter? Well, here we go. Oops, other way. I was just about to complain about my computer, but now I mess up. So there you go. That's what I get for complaining about my computer in my mind. All right, so here we go. Almost there. All right, here it is. So when there's a shortage of oxygen, then electrons clog the pumps on the ETC. So if the pumps are clogged, then the full baskets can't drop off their electrons. So eventually the cell has only full baskets and no empty baskets, right? So here we'll go to our overview page. Right, so you'll notice, right, all of these full baskets, okay, so NADH, NADH, and FADH2, all of those go to either pump 1 or transport protein number 1. Well, if there's so many electrons here that pump 1 or transport protein number 1 aren't accepting electrons, then all of a sudden the full baskets just kind of fill up and they're just kind of standing there waiting. Right? It's like the shopping cart line at Costco. Your shopping cart is full and you're just standing there and you're waiting, and it's like all of the checkers have gone on break all at the same time. Or their machines aren't working, or they're just clogged. You're just standing there, you're waiting. Well, eventually, if that goes on long enough, you're not going to have any empty shopping carts in the front of the store. They're all going to be full, waiting at the checkout line. Right? That's a fairly good analogy that I just happened to make up on the spot. Thank you, Lord. So we have only full baskets. Right? So now if we continue, there we go. So now we have only full baskets and no empty baskets. Well, if we have no empty baskets, then we can't do glycolysis or Krebs. All right, so this is where you have to understand my drawings a little bit. All right, I know it's difficult, but we're gonna walk through it. So this is a process. This is a chemical reaction. It's like a recipe, right? So for this to happen, you need glucose and you need ATP because both of those things feed into it, right? This is the product. It makes ADP. Okay, for this reaction to happen, you need this compound right here, which shall remain nameless because I don't want to confuse you. You need ADP and you need NAD+. If you don't have any one of these, then this reaction can't happen. It's like trying to make chocolate chip cookies without flour, right? If you don't have flour, or trying to make chocolate chip cookies without chocolate chips. If you don't have flour, you can't make chocolate chip cookies. If you don't have chocolate chips, you can't make chocolate chip cookies, right? It just takes one to mess up, one lack of one ingredient to mess up this whole entire thing. So we don't have any NAD+. So this can't happen, right? And we also use NAD+, we also use empty baskets in the Krebs cycle. We use a lot of empty baskets in the Krebs cycle, right? No NAD+, this can't happen. Okay, no NAD+, FAD+, step 5 can't happen. If we don't have any NAD+, step 4 can't happen. No NAD+, step 3 can't happen. So the whole entire thing just goes absolutely pear-shaped. It just gets shut down and there's no recovery. All right, so what does that mean for the cell then? Well, if there's no glycolysis or Krebs, then we don't have any ATP. Okay, well, that's kind of important, right? If we have no ATP, then we have no energy. That's kind of important. If we have no energy, then the cell dies, right? And if enough of our cells die, then, yes, you guessed it, we die. And that's not very good, is it? So God, in his infinite wisdom, has created a secondary pathway, right? So cellular respiration is the main pathway by which our cells get energy under ideal conditions. But if for some reason we're working so hard that our lungs and our bloodstream can't carry enough oxygen to our muscles, then God has created a secondary pathway called fermentation. And it actually operates slightly differently in muscle cells and in other cells like bacteria and yeast and stuff like that. So we're going to look at muscle cells first. So in muscle cells. In muscle cells. So fermentation, we're going to start with pyruvate, right, which is the product of glycolysis, right? And then pyruvate is going to, it's just one step, is going to be broken down and it's going to make lactic acid. Lactic acid. So part of that is taking a full basket, NADH. I'm sorry, I said broken down before. I meant to say built up. Because we're going to take a full basket, we're going to get rid of our hydrogen, we're going to get rid of our electrons, and we're going to make, oops, NAD plus. We're going to make an empty basket. All right. This right here is the whole goal, is the whole point of fermentation, because this is then going to go to glycolysis. And that's going to provide the ingredient that glycolysis needs to run. All right, lactic acid now is toxic, and so our cells have a way to get rid of that, and that's part of what makes your muscles burn and what makes your muscles feel fatigued after a very, very intense workout, such as like your max on bench or sprinting or planking or something like that, because it takes a lot of muscles to maintain that posture. So that's what happens in muscle cells, right? And Mrs. Lindsay is going to come back and talk about that in her videos. But then in yeast, something slightly different happens. In yeast, it's going to start with pyruvate, right? And it's still just kind of one step, right? But then here it's actually broken down. This is the one that's broken down. So pyruvate is going to be broken down, and it's going to lose a CO2, right? And then also it's going to do the same thing. It's going to take a full basket, NADH, and it's going to make it into an empty basket, NAD+, right? And then again, that's the whole goal. So that goes to glycolysis. Oops, GLY. And then that's going to make alcohol. Right, so fermentation yeast, if you mix a little bit of yeast with some grapes and right temperature and right other ingredients and stuff like that, then you get wine. Right? If you mix yeast with hops, then you get beer. If you mix yeast with a bunch of other stuff, then you get hard alcohol. Agave makes tequila and different things like that. Or we also use yeast when we make bread, right? And this is really, really cool. So if you've ever done any bread making or any baking, you know that you put the yeast in and you mix it all in and you make the dough and stuff like that, and then you let it sit. And what does it do when it sits? Well, it rises. Why does it rise? Well, the yeast inside the bread dough, it doesn't have any oxygen, and yeast have different energy requirements than humans. So yeast are totally fine living their whole entire lives without oxygen. We can only operate for like 90 seconds without oxygen at full capacity. So the yeast makes CO2. This CO2 is what makes bread rise. Right, so CO2 makes bread rise. He says, Mr. Leo, okay, so if this process is going on when I make bread, then shouldn't I get drunk when I eat bread? Isn't it making alcohol? Well, it is making alcohol, but then after you let it rise, then you put it in the oven and you cook it, and the alcohol evaporates. The alcohol bakes off. Right, so CO2 makes bread rise. This is also used in fermentation to make alcohol. And the next video, we're going to talk about how to make you a better athlete, how to use these two processes, cellular respiration and fermentation, and to take those into account and to make you a better athlete and to have a better workout. So this has been Mr. Leo. Today we looked at the process of fermentation, why we need it, and the process, how it happens. Like I said, thanks so much for watching, and I will see you on the flip side.